This is the Translational Research in Clinical Oncology course. And we call it TRACO for short. So I direct it. My name is Terry Moody. I'm with the Center for Cancer Training at NCI. My office is up at Shady Grove, but if you need to contact me, the best way is through email. And so our organizing committee, we have Erwin Arias. He directs the Demystifying Medicine course. So this is done in the spring on Tuesday afternoons. And he goes through numerous diseases, whereas we focus especially on cancer. And Jonathan Wiest is the director of the Center for Cancer Training, and we thank him for his support for this course. And then we also have Lubia Wojciechowski and Farazia on the organizing committee. So TRACO, usually it's Mondays 4 to 6, but we couldn't get the room every Monday. So today it's on a Tuesday due to the holiday yesterday. And September 17th and 27th, it's also on a Tuesday. And then there's one date in October where it's on a, actually it's on a Thursday, September 17th and 27th. And a Thursday, one date in October, I think it's October 11th. And then the rest of the time it's on Monday. So 9 of the 13 lectures are Monday afternoons, 4 to 6, in this building. And then we videocast the lecture live. So there's probably about 30 people or so watching it live on their computers right now. We also video conference it to NCI Frederick. And there's probably about 15 people or so watching it live there, and we welcome the people at NCI Frederick. So NCI, we're so big, about a quarter of our staff is up at the Frederick campus. The rest of it's in Bethesda and up at Shady Grove. So my focus is especially lung cancer, and then we have several other lung cancer lectures. Halbin Chen lectures on small cell lung cancer September 13th. And then Eva Szabo lectures on the non-small cell lung cancer on November 5th. So you'll get a good picture of that. And the reason being, lung cancer is the big killer in the US. We have about 150,000 people dying each year from lung cancer. Overall, there's about 500,000 people in the US each year that die from cancer in total. The other big killers are breast cancer, which Farazia will lecture on October 22nd. We have prostate cancer, which Ravi Madan lectures on September 13th. And then lung cancer, it's also very difficult to treat. So the other ones that are difficult to treat are ovarian cancer, which Tina Annunziata will lecture on September 17th. And then we have prostate cancer. Hussein Perez will lecture on that on December 3rd. And then brain cancer is also extremely difficult to treat. And you're all familiar with John McCain. Recently had his funeral this weekend here in DC. He died from glioblastoma. So with cancer then, there's really 4 ways to treat it. If you're stage 1 early cancer, you can just do surgery. If it's stage 2, you can do, and stage 3 and stage 4, you can do chemotherapy. And we have Yves Pommier lecturing on October 29th on the topoisomerase inhibitors, their chemotherapeutics. And then we have radiation oncology. Elizabeth Nichols lectures on that October 11th. And then the immune system is becoming increasingly important to treat cancer. So we have a new lecture this year on CAR T-cell therapy for leukemias. And then we have immune checkpoint inhibitors by Stephanie Goff. And these are very effective in melanoma, and they're also quite good to treat non-small cell lung cancer. So you see we cover many different types of cancer throughout the course. And then a key factor is precision medicine, which is lectured by Kurt Harris. And precision medicine, all of the patients here at NIH, they undergo genomic analysis. And we have Jun Wei talking about genomic analysis here. And depending upon what they see is mutated, that in turn dictates the therapy. So for cancer, routinely we have dozens of mutations, and some of the mutations are key driver mutations that play a big role in the proliferation of the cancer. And another mutation that's very common is the KRAS, and Ji Luo will be talking about that. KRAS mutations are very prevalent in pancreatic cancer. Also, some types of lung cancer have KRAS mutations. And we have a program up at NCI Frederick that's trying to come up with new therapies for patients that have KRAS mutations. And finally, we end with nanotechnology. So this involves developing better ways to treat cancer, minimizing the side effects of cancer therapy. So the course then, the registration is online, and we have over 120 registrants at this point. But, you know, many people watch online, and many attend the class as well. So, and then a unique thing of this, it's not just an academic course, but you can do additional things, such as you can visit tumor boards, you can visit grand rounds. Those are usually held Friday at noon in Lipset Auditorium. And you can visit technology and core facilities. So I'm making arrangements for this now, and once I've made all the arrangements, I'll be sending you emails, and you can sign up and visit the tumor boards or the core facilities if you so desire. It sort of adds additional perspective to the NCI component. And finally, at the end of the course, you can get a certificate of completion. So what we do is we have one question from each lecture. It's multiple guess. And then if you get 70% of the questions right, you get a certificate of completion signed by the director. So routinely, we have about 50 people from the class getting the certificate at the end. And we post it online after the last lecture, which is December 3rd. So a key thing here then is the video cast. If you can't attend a lecture, you can always watch it on video cast. 2 days after the lecture, the video cast is then archived. So then you can call it up on your computer and watch it anytime. Okay, are there any questions at this point about the organization? Okay, then we'll proceed to the introduction. So here we see that in the US, there's about 1.3 million cases of cancer diagnosed each year, and about half of these people die. There's about 550,000 deaths. We mentioned before that the 4 big killers are lung cancer, over 150,000, colon cancer, about 50,000, breast cancer, 40,000, prostate cancer, about 30,000 deaths. So these 4 comprise about half of the total deaths. And lung cancer, you see most of the people that get it die. And lung cancer is very complicated. It takes 20 to 30 years to develop. So there's numerous gene mutations. And it's very difficult to treat, but we're starting to make some inroads. In the 2000s, we developed tyrosine kinase inhibitors for receptor tyrosine kinases. And in the 2010s, we've developed immune checkpoint inhibitors. And these are effective in about 15 to 20% of the non-small cell lung cancer patients. So we have optimism at this point. For colon cancer, about a third of the people that get it die, so there are some treatments, such as chemotherapy, that are fairly effective. Breast cancer, you see about 20% of the people that get it die. So breast cancer, we're having more and more therapies developed all the time. And here at NIH, if a breast cancer patient comes in, they're very confident that they'll be able to stop the cancer and keep that patient alive for many years. And for prostate cancer, it's the same thing. So basically what's happened is the total number of deaths now has leveled off in cancer. Our treatments are getting more and more effective, and the number of cancer survivors is readily increasing. We now have about 15 million cancer survivors still living. And each number, that increases by 1 to 2 million. So our therapies are getting better and better for cancer treatment. So we've gone through the big four. There's other types of cancer that we have to worry about, such as pancreatic cancer, very difficult to treat, lots of KRAS mutations. Ovarian cancer, very difficult to treat. And we mentioned brain cancer is also very difficult to treat, especially glioblastoma. And we'll be discussing leukemia. That's more readily treatable, especially by CAR T cells. You'll have a lecture in that later. And stomach cancer, not so prevalent in this country, but in Asia, it's very common. So it has to do with the foods they eat there. They're highly pickled, and as a result, they get a lot of cancer of the stomach in Asia, China, and Japan as well. And then liver cancer, it's very big in China again, but the US, it's more treatable. And then we have esophageal cancer. That's associated with cigarette smoking. So cancer risk, alcohol. So if you drink too much, it can result in liver cancer and especially esophageal cancer. Asbestos. So this is in the walls of buildings, and many old buildings still have asbestos in the walls. I used to have an office in building 31, and at the top of the wall, there was a little notice that said, "Danger, asbestos in walls." As long as the wall's solid, that's fine. But if the wall breaks open, then that asbestos comes out in the air, and you can get mesothelioma of the lung. Diet. So we mentioned that you want to eat foods that are low in fat, such as fish. The fat increases the risk of colon cancer. There's familiar factors, genetic factors, such as breast cancer. You can have BRCA1, BRCA2 mutations. You're at high risk of getting breast cancer. Also, there's familial components to colon cancer. Hormones, we have estradiol in women contributing to breast cancer growth. We have testosterone in men contributing to prostate cancer growth. Obesity. So if you have a big body mass index, and throughout the world the body mass index is gradually increasing, you're at high risk for colon cancer. Ionic radiation, such as the atomic bombs. In Japan, they released lots of radiation. There was lots of leukemia. Tobacco, if you smoke cigarettes, you're at increased risk for throat cancer, as well as especially lung cancer. Ultraviolet radiation, if you live in the southern United States, especially Florida, Arizona, you're at high risk of getting melanoma. And finally, there's viral components, such as today, John Schiller will be lecturing next. He'll be talking about cervical cancer, which is viral in nature. So in terms of smoking, there's 45 million current smokers and 45 million ex-smokers. When you stop smoking, your risk of heart disease goes down almost immediately, but for lung cancer, it takes about 10 years of not smoking to get back down to normal lung cancer risk. And it's difficult to quit smoking due to nicotine addiction. So in the smoke, we have many chemicals, such as polyaromatic hydrocarbons. And then we have 4-methyl nitrosoamino 3-pyridyl butadienone, NNK. It has additional things such as ethyl carbamate. This is common in varnish for the floors. And then it has metals such as chromium. And finally, hydrazine, which is rocket fuel. So you see there's numerous components in cigarette smoke that can lead to lung cancer. And in particular, the polyaromatic hydrocarbons, benzapyrene, is converted to a metabolite BPDE, which then forms EDX with the DNA, leading to genetic mutations, C to G to A to T to A mutations in the DNA. And especially the genes for P53 and KRAS are frequently mutated in lung cancer. So here's benzapyrene. You see it's a chemical with 4 aromatic rings. And then we have some hydroxyl groups coming off of this 6-membered ring. And basically what happens is the benzapyrene can be oxidized by enzymes, P450 enzymes, epoxide hydrolase, leading to BPDE, which can then form DNA adducts and mutate the DNA. So cytochrome P450 enzymes, they catalyze oxidation of the carcinogen, of the benzapyrene to the carcinogen, increasing its water solubility, and the carcinogen can then be metabolized by phase 2 enzymes and finally excreted in the water, in the urine. So DNA is mutated if the rate of carcinogen activation exceeds the rate of carcinogen detoxification and or DNA repair. DNA adducts as well as intra and interstrand DNA crosslinks are removed by nucleotide excision repair. So we have the carcinogen then, it gets the chemical in the smoke, it gets oxidized to the carcinogen, and then that can lead to DNA mutations. Now, normally P53 mediates the G1 to S phase of the cell cycle. It drives programmed cell death after DNA damaged. It's increased along with P21 after DNA damage, and phosphorylated DNA induces expression of the FACS, which then causes apoptosis of the cancer cell. So the mutations in the P53, they're not just random, but they occur at specific areas where there are CpG-rich codons, and this is exon 5 and exon 7 and exon 8 of the P53 gene. And there's an excess conversion of G to T transversions in smokers relative to non-smokers. So here you see the P53 gene, and we see there's certain areas where there's lots of mutation, particular codons 157 and 158, 245, 248, 249, and 273. So then in the cell cycle, normally the cells are resting in the G0 phase or G1 phase. And when they undergo DNA replication, they enter the S phase. Then they go to the G2 phase where they get ready for mitosis in the M phase. And here we have 2 daughter cells being derived from a parent cell. So cancer cells, they basically try to grow as fast as they can. And P53 basically stops the G1 to S transitions, but when P53 gets mutated, then the cell readily goes from G1 to S. And so we have various enzymes then that mediate the cell cycle progression. And so we have cyclin D. It can combine with cyclin-dependent kinase 4, 5, and 6. We have cyclin E. It can combine with cyclin-dependent kinase 2. And then in the S phase, we have cyclin A combining with cyclin-dependent kinase 2. In the G2 phase, we have cyclin A combining with cyclin-dependent kinase 1. And in the M phase, we have cyclin B combining with cyclin-dependent kinase 1. And with cancer cells, basically this is going as fast as it possibly can. So cancer cells duplicate every day. So then with tobacco smoke, after 10 years of smoking, the normal lung tissue can undergo hyperplasia and metaplasia. After 15 years, dysplasia can result. After 20 years, a carcinogen in situ can form. And after 25 years, a malignant cancer can develop. So here we see a little cartoon. In red, we have normal cells. And with hyperplasia, these cells start to proliferate and increase in number. In dysplasia, these cells become disorganized. And then when we have a carcinoma in situ, there's some cancer cells, but they're still retained to the normal tissue, which in this case would be the lung. And then when we get a malignant cancer forming, the cancer cells go from the lung to other organs. And with cancer, they go to the lung, lymph nodes, brain, and liver. And essentially, these cancer cells, as the cancer progresses, it can just overtake the normal organs. And in a dying patient, if you were to cut into their liver, you would see almost all cancer cells and very little liver remaining. So this is the normal lung, and we have a bronchus here. And this is the alveoli where you breathe. And there's cilia on the top here. And this exchanges gas. You breathe in oxygen, and you exhale CO2. But then we now see hyperplasia. The number of cells is increasing in the alveoli, and the cilia are still there, breathing. But as we go to metaplasia, now we see the alveoli are becoming very disorganized, and the number of cilia are greatly decreasing, so you're going to have trouble breathing. And then when we have a carcinoma in situ, this is what it looks like. And then when we have a malignant melanoma, it sort of forms a cluster of cells, and they secrete various entities from these clusters. So again, the cartoon in white here, we have normal cells. When we have cancer initiated, we have some cancer forming, but it's still restricted to the local area, in this case, the lung. And we then still have a carcinoma in situ, but then it becomes malignant, and the red malignant cells can undergo metastasis through the blood and the lymph nodes to other organs. So as tumors grow, they start out in a certain organ, like the lung. And then as the tumors become big, they need blood vessel formation, angiogenesis, because they'll start to die if they don't get enough oxygen and nutrients. So what happens is the host then sends blood vessels into the tumor to give it additional nutrients, and then the cancer can continue to grow, and it can migrate and invade other organs through the process of metastasis. So in lung cancer, genetic abnormalities, we've mentioned we have P53, it gets mutated. And then another thing that gets mutated is the retinoblastoma gene. And it can get mutated or silenced. And these are essentially tumor suppressor genes. But then we have oncogenes. They get mutated. And when they're produced in excessive amounts, they stimulate the tumor growth. And in lung cancer, two of the big ones are the EGF receptor and the ERB2 or HER2 receptor tyrosine kinase. We mentioned cyclin D1. That's something that stimulates the G1 to S transition in cells, stimulating their growth. And c-MYC is also an oncogene in the nucleus, and when it gets overexpressed, it stimulates growth. So we see an example here then of 7 things that get mutated or silenced. We mentioned before that in lung cancer, usually there's about 50 genes that are mutated. So these are the receptor tyrosine kinases. They're large proteins in the membrane. We have the EGF receptor. It binds growth factors on the extracellular surface, the 1 and the 3 domain. And then when it binds, it causes a conformation change in the receptor tyrosine kinase that's in the cytosol. And then this can phosphorylate various proteins. So we see also the insulin receptor or IGF-1 receptor. It's also got a binding site on the outside and a receptor tyrosine kinase domain on the inside. But the EGF receptor is a monomer. This is essentially a dimer. And then others include the NGF receptor, PDGF receptor, FGF receptor, VEGF receptor, which stimulates angiogenesis, and the F receptor. And you see with these, they all have a receptor tyrosine kinase domain, but what differs is the extracellular domain. Here there's lots of immunoglobulin-like domains in the NGF, PDGF, FGF, and VEGF receptor, and there's also one in the F receptor. So these receptor tyrosine kinases then, they're what really gets the cancer growing. And so here we see the EGF receptor. Well, there's lots of ligands that can activate it. EGF receptor, TGF alpha, this is the one that the cancer cells like to make. Amphiregulin, beta-cellulin, heparin binding EGF, that's another one that cancer cells like to make, and epiregulin. So the cancer cells then they can make at least 2 different ligands that can activate the EGF receptor. Surprisingly, the ERB2 receptor doesn't have any ligands. And what happens is the EGF receptor, when it's activated, it can make itself a dimer. If it's 2 EGF receptors, it's a homodimer. But it can also make a heterodimer. So the EGF receptor can interact with the ERB2 receptor, causing its phosphorylation and turning on its kinase activity. So these growth factors then and the receptors, they don't work singularly. They work in concert with one another to stimulate growth. So here we have the EGF receptor. We've got 621 amino acids in the extracellular domain. There's a 24 amino acid transmembrane domain. And when the EGF receptor binds TGF alpha, it causes a conformation change and turns on the tyrosine kinase activity. We have 541 amino acids in the cytosol. And in particular, a lysine at 721 binds ATP, and it transfers the phosphate then to tyrosine amino acid residues on substrates. And then the EGF receptor can get phosphorylated at 4 different tyrosines. So here we see that EGF binds with high affinity, so does TGF alpha, and there's monoclonal antibodies then that have been made that also bind with high affinity to the EGF receptor and block it. And here we see when the EGF receptor gets activated, we see a strong band here at 170. That's the EGF receptor itself. But also, this is phospholipase C gamma that gets phosphorylated, and PI3 kinase get phosphorylated. So when the EGF receptor is activated, then numerous things get tyrosine phosphorylated. And this leads to growth increases. So a very curious thing about 15 years ago, they found that the EGF receptor was mutated in lung cancer, but not other types of cancer. And so in particular, this mutation at amino acid 858, where leucine replaced an arginine, or at 719, where glycine replaced a cysteine, this then turned on the tyrosine kinase activity to stimulate growth, even in the absence of ligand. So then they developed these small molecule tyrosine kinase inhibitors to block the EGF receptor. And a little later on, we'll have a lecture on small molecule inhibitors. And for the EGF receptor, gefitinib became a very popular drug in the US, and erlotinib became a very popular drug in Europe. They both block the EGF receptor. And so in patients then, it was found that these were very effective in patients with EGF receptor mutations for about 1 year. But what can happen then is what's called resistance. And so the cancer knows that the EGF receptor is being inhibited, so it tries to figure out a way around it, and it mutates into something else. And so the EGF receptor, then it can undergo additional mutations and bypass the effects then of the gefitinib and the erlotinib. Now, normally the EGF receptor, when it binds, it can then stimulate a growth pathway. And this is called the RAS, RAF, MEK, ERK pathway that leads to cancer cell proliferation. And we mentioned that KRAS is mutated in about 20% of the non-small cell lung cancer patients. So KRAS can be turned on if you have the mutation in the cancer cell. So with RAS then, RAS is a GTPase, and what it does is it converts, it binds the GTP and activates itself. And there's RAS mutations in codon 12 whereby it's turned on. And when RAS gets turned on, then it works on RAF, which is downstream. And RAF gets mutated in melanoma patients. We have the valine at 600 replacing a glutamate. And so they found a tyrosine kinase inhibitor for this, PLX 4032. And this kinase inhibitor, when given with patients, melanoma patients that have the mutation, they get an 81% response rate. And all's very good for about a year or so until the cancer mutates into something else. And so basically what cancer is it's a moving target. So the clinician finds one drug that works, and it may work for a year or so, but then the cancer can mutate into something else. And then the clinician has to find something else that's going to stop the proliferation. So downstream of RAS, we have MEK. And so MEK is an enzyme, and they have a good inhibitor for this, trimetinib. And another one is selumetinib. And so they're combining this with chemotherapeutic drugs, docetaxel, in KRAS mutant non-small cell lung cancer patients. And downstream of MEK, we have ERK. And so ERK gets tyrosine phosphorylated, and when it gets phosphorylated, it can go into the nucleus and alter transcription of genes, such as FOS, JUN, or MYC. And this all leads to cancer proliferation. So here's a little cartoon again. We have the EGF receptor heterodimer forming, and then it activates the RAS, which then activates the RAF, and then the MEK gets phosphorylated, the ERK gets phosphorylated, and ERK can then enter the nucleus and alter gene expression, leading to proliferation. So here's a computer diagram then of what goes on when you have an EGF receptor heterodimer forming. The EGF receptor, we have a monomer here, and then it forms a dimer that's inactive. And when the dimer gets activated, it sort of twists around itself. And then it's biologically active, and it causes increased proliferation. So another thing that happens is not just proliferation, but when the EGF receptor gets activated, you inhibit apoptosis of cancer cells. And so the pathway for this is through the PI3 kinase, which can then activate the AKT, which activates the mTOR. So when the PI3 kinase gets activated, it causes conversion of the lipid PIP2 to PIP3. And then this is what basically leads to the decreased apoptosis of the cancer cells, the increased survival. So PI3 kinase, it's mutated in breast cancer and brain cancer and colon and stomach, not so much in lung. And when it gets mutated, then you have increased activity of the PI3 kinase, leading to increased survival of the cancer cells. So the PI3 kinase, it can be metabolized by this enzyme, PTEN. But sometimes PTEN can get mutated in about 13% of the breast cancer patients. So if you lose PTEN, then you have lots of PIP3, leading to increased cancer cell survival. So downstream of PI3K, we have AKT, and AKT gets phosphorylated, and when it gets phosphorylated, then it can phosphorylate BAD and caspase 9, preventing apoptosis of cancer cells. And AKT is mutated in some breast and ovarian cancers, not so much elsewhere. When AKT is activated, downstream of that is mTOR. So we have two then cycles that are very important, one leading to proliferation, the ERK pathway, and one leading to increased cancer survival. That's the PI3 kinase AKT pathway. So in lung cancer, KRAS is mutated in about 25% of the cells. EGF receptor is mutated in about 15% of the cells. AKT is another enzyme. It's mutated in about 5% of the cells. And HER2 is mutated in about 2%. And you see there's other things that have been identified as well. But we see here the largest component is the unknown. So even though we've made a lot of progress in the last decade identifying many additional things that are mutated in lung cancer, besides KRAS, we've still got a long ways to go. There's about 40% of the things that they still think they have to discover to get a genetic feel for lung cancer. But we're making progress, so it's a very exciting time. And this you probably can't see, but it illustrates precision medicine in lung cancer. This is a protocol that was done at the Center for Cancer Research on lung cancer. And so they did the genetic analysis, and the first thing they look for is EGF receptor mutation. And if it's mutated, then they put the patient on erlotinib, the tyrosine kinase inhibitor. The second thing they look for is RAS mutation. If they see that, they then put them on a MEK inhibitor. The third thing they look for is a PTEN mutation. If they see that, then they use a PI3 kinase inhibitor on the patient. The fourth thing is they look for ERB2 mutation. If they see that, they treat the patient with lapatinib. And the next thing they look for is a c-KIT mutation. If they see that, they put the patient on sunitinib. So this is precision medicine. The mutations that they observe then dictates what therapy the patient's going to get. Okay, so this is a cartoon illustrating that cancer cells don't go by receptor tyrosine kinases alone. We've mentioned the EGF receptor and how it can form heterodimers with HER2. But it turns out G protein coupled receptors can also cause tyrosine phosphorylation of the EGF receptor in HER2 through a process called transactivation. So here we have the G protein coupled receptor causing second messenger production. The second messengers then come and activate a matrix metalloprotease enzyme. And what it does is it cleaves TGF-alpha from the plasma membrane precursor. The TGF-alpha can then bind to the EGF receptor, leading to tyrosine phosphorylation of the EGF receptor and HER2. And then downstream, it causes phosphorylation of the ERK. And this whole complicated process, when we activate these G protein coupled receptors, we see phosphorylation of the EGF receptor in 30 seconds. It's extremely rapid. We see tyrosine phosphorylation of ERK within a minute. So these processes are super fast in the cancer cell. Okay, and we mentioned about the resistance. About 50% of the people develop resistance to erlotinib or gefitinib after 1 year. And this can be due to secondary mutations of the EGF receptor, such as we have at position 7090 threonine replacing a methionine. Or it can be other things, such as activation of the c-KIT or other factors. So another big example of a tyrosine kinase inhibitor is with CML patients. So here you have a blood disease. And it results because of a translocation, not a mutation, but a translocation. And so we have BCR-able tyrosine kinase activity then is stimulated as a result of this translocation. And this is inhibited by the tyrosine kinase inhibitor Gleevec. So we have chromosome 9 and chromosome 22 forming a translocation. So here you see from chromosome 22, we have the N-terminal combining with the C-terminal of chromosome 9. And so now we have the BCR-able gene, and then it's translated into the BCR-able fusyl protein, which then stimulates growth. So in the 90s then, they found Gleevec could inhibit this receptor tyrosine kinase. So here's the chromosomes, chromosome 9 and chromosome 22. You see after the mutation, the translocation, chromosome 9 becomes bigger, chromosome 22 becomes smaller. And then here's our product right here, the BCR-able. So translocations then are also very important in cancer. It's not just mutations. And another factor that we'll talk about later in the course is epigenetics and how that silences genes, and that plays a role in cancer formation as well. So the toxicity, though, of the Gleevec was nausea, vomiting, edema, and rash. So all of these tyrosine kinase inhibitors, you try to use them, but you have to watch out for side effects. So you want to use as low a dose of tyrosine kinase inhibitor as you possibly can. So we see initially in Gleevec, after a year, 53 out of 54 patients responded. But over a 5-year period, 89% of the patients treated with Gleevec had progression-free survival, but some of them became resistant to the drug. So here we see Gleevec. It's a small molecule inhibitor like gefitinib or olatinib. And when it binds in purple to the BCR-ABL, then there's a loss of enzymatic activity, no signal transduction, and the leukemia levels are greatly reduced. So after a 5-year period, 17% of the patients initially sensitive to gefitinib became resistant, and they found that the BCR-ABL gene had mutated. And in particular, at position 315, a threonine replaced an isoleucine, impairing Gleevec's ability to bind to the receptor. So what do the pharmaceutical companies do? They synthesize the next generation of BCR-ABL inhibitors. And in this case, it's ponatinib. In lung cancer now, we've had 3 generations of EGF receptor tyrosine kinase inhibitors. So we keep the chemists busy, and the problem is cancer is a moving target. The drugs that will initially work, but then the cancer can become resistant by mutating into something else. So that's the problem with cancer. It's not static, it's always changing. So then in terms of tyrosine kinase inhibitors, we mentioned that Gleevec was very effective for BCR-ABL. Another one is dasatinib. For breast cancer, they use HER2 inhibitors, especially Herceptin, which is an antibody. Lapatinib is a tyrosine kinase inhibitor. For melanoma, there's BRAF mutations. They use PLX4032, a tyrosine kinase inhibitor. For gastrointestinal stromal tissue, they have mutations of c-KIT. They use imatinib as well as sunitinib as a receptor tyrosine kinase inhibitor. And with non-small cell lung cancer, you have EGF receptor mutations. The tyrosine kinase inhibitor is gefitinib as well as erlotinib. Okay, so now we do the wrap up. So practical steps to prevent cancer. This is why you're all here. This is what you want to learn. First thing, check your house for radon. So I was educated in California. I came to Maryland, started working at NIH, bought a house up on a hill in the country. And it turned out that in many of the hills and mountains in Maryland, there's rock in the mountains, and radon comes out of the rock and gets into the basement of the house. So when I went to sell the house, I was told, we can't sell it until you put in a radon pump. If you lived in the basement of that house, it would be the equivalent of smoking 2 packs of cigarettes a year. Well, we all lived in the upper floors, but you know, to sell the house, then we had to put in a pump, and basically it just pumps the air out of the basement into the environment. Dilution is the solution. Okay, check your house for asbestos. So we mentioned that it's fine if it's in the wall, but if the wall starts to break down, then you're exposed to the asbestos, and that's not good. Take precautions at your workplace. I told you the story of the asbestos in the walls of Building 31. So many of the old buildings here at NIH, that's what they have in the walls, asbestos. Check your community water system. So you don't want to drink polluted water. There's an example in Buffalo of people drinking polluted water, and then they get increased risk of getting cancer. Avoid breathing polluted air. When I was a grad student, I went to school at Caltech, and there's ozone all over the Los Angeles atmosphere. Every day, we were in Pasadena around 1 o'clock. the ozone from LA would blow into Pasadena, and you couldn't breathe. You felt like your lungs were burning. And then by 3 o'clock, it had blown over to Riverside. So they say in LA, for every 2 years you live there, you take about a year out of your life just from breathing the bad air. Okay, protect your skin. So we mentioned with melanoma, that's a big problem. And so you put on sunscreen to try to prevent the exposure of the ultraviolet radiation on your skin. And basically what happens is in the earth, we have so much polluted air now that more ultraviolet light comes in from the sun. Don't breathe smoke. One of the ways they found lung cancer was in England. The people who swept the chimneys got lung disease. And it's just the soot in the chimney. They were breathing it all the time, got into their lungs, they got cancer. Exercise daily. I love to ride the bicycle in the country, but unfortunately this year I had knee surgery, so I'm out of the race. Maybe next year. Avoid pesticides. There's sort of a general trend where many farmers who use pesticides, they have increased incidence of prostate cancer. Eat fruits and vegetables. These have lots of reductants. We went in great detail about showing you how a chemical was oxidized into a carcinogen. Well, if you have lots of reductants, then it's harder to get things oxidized to form carcinogens. Reduce red meat consumption. You want to eat fish with omega-3 fatty acids. The fat leads to increased incidence of colon cancer. So you want to eat fish, minimize fried foods that has fat in it as well. Drink alcohol in moderation. You don't want to overdo it and destroy your liver or get esophageal cancer. Avoid unnecessary x-rays. And then you want to reduce infections. About 18% of the cancers are related to infections. So here's a few references, and basically that's it for me. I'd be happy to answer any questions. Yeah, you want to basically reduce oxidants in your environment, such as if you go to Japan, you see the water there, they advertise it as having reductants. And then if you come to the US, they sell you water in the airports and they advertise that they have oxidants. I would much rather drink the Japanese water than the American water. So basically, like for lunch, I eat lots of fruits and vegetables, and those have lots of reductants in them. And so that's very good. And another thing you can do is take lots of vitamins, because that erases the effects of a lot of the oxidants. So oxidants, they basically increase your aging, and you want to reduce that process because it also reduces your risk of carcinogens. Yeah, there was one study in lung cancer that was done in Europe, and they found that giving the vitamins was actually slightly increasing the risk. They don't know why, but then the trial was stopped. So basically, what happens is diet has a lot to play with cancer. And the diets vary dramatically from one country to another. They vary from person to person. So when you're trying to prevent cancer, you want to take that into consideration is, what you're eating in the long term, that's going to have a long effect on, how well you live and how long you live. So you don't appreciate that when you're young, but as you get older, you do. Anything else? Yes. Question is really about P53 and its mutations. And so what happens in cancer is oftentimes one gene gets mutated, but you have two genes of everything. And then the trick with cancer is once the first gene gets mutated, then the second gene gets inactivated. And then it's like you don't have any p53 that's functional. And so that's sort of the problem. And with cancer, this happens a lot, whereby you have a mutation, and then the other gene gets inactivated, and then that's a big problem. And, you know, you might think, well, you could just do gene therapy, but that hasn't proven effective. I wish it was, but it hasn't really done much. Okay, so we'll move on now to the next lecture, which is John Schiller. So John's a distinguished investigator. He's in the Laboratory of Cellular Oncology, and he graduated from the University of Wisconsin-Madison. And then in 1982, he received a Ph.D. from the University of Washington in Seattle. And then he came to NIH, first as a senior staff fellow, and as a senior investigator, and he became chief of the neoplastic disease section in 1998. And he's received numerous awards for his contributions to papillomavirus and the molecular biology and HPV vaccine development. And in 2017, he received the Lasker Award. Congratulations.
Thank you. Thanks for being here. It's good to be talking to you. It's the first lecture. There's like the most people here. Usually after a while, people start doing it online. But so it's nice to actually have you here today. So on your list, it says that I'm going to be talking about cervical cancer. Well, it's not entirely true. What I'm really going to be talking to you today is about one of the preventive measures that Terry just mentioned, and that's preventing infections that cause cancer, and particularly HPV, which is responsible for about 5% of all cancers. And the reason he says cervical cancer is because that's the predominant cancer worldwide, as you'll see in a minute. So what I'm going to be talking today about is there a pointer?
I was just hitting the arrow.
This one?
That's just like the right.
Where's the? Oh, right here. Okay, I was playing too high. Okay, so what I'm going to be talking to you about, I would tell you a little bit about the association between cancer and okay. Yeah, it's a little bit better. So I'm going to be talking to you about the association between HPV and cancers, and then talk about some vaccines that we developed. Hopefully you've heard about these vaccines, Gardasil and Cervarix, to prevent HPV infection, and talk about some of their efficacy and effectiveness, and then talk to you about some of the key implementation issues that we've run into in trying to make this an effective public health intervention program, and then talk a little bit more of the molecular biology of why they work so well. These actually work way better than we ever imagined they would work. It's been really interesting to try to figure out why that's the case. And then finally, if I have some time, tell you a little bit about a new cancer therapy project that we've developed that's sort of grown out of our findings about the basic biology of this virus that leads us to hopefully develop broadly applicable cancer therapy strategy. So again, the reason when we talk about cervical cancer and HPV is because the vast majority worldwide of HPV-associated cancers, about 5% of all cancers, are cervical cancer. And then, but you can see that there's these other cancers that are also associated. And I should point out that 70% of these cancers for the cervix are caused by one of two HPV types, 16 and 18, whereas it's even a greater percentage for these other cancers. So obviously these are the two types that we really want to focus on when we think about vaccines to prevent cancer. Now the story in terms of burden of disease is a little bit different in countries like the United States. You can see cervical cancer is still a large fraction, but it's only about half. And because of oropharyngeal cancer being predominantly in men, the burden of male HPV-associated cancers is higher in the developed world than in the developing world, and that's mainly for two reasons. One, we have effective Pap screening programs, which has reduced cervical cancer by about 80%. We don't have those types of screening programs for oropharyngeal cancers because the premalignant lesions have not been identified. And the other thing, there's been what's been considered an epidemic of HPV-associated oropharyngeal cancer with an over twofold increase in a couple decades, and it continues to go up, probably because of differences in sexual practices over the years. Now, the virus that we're talking about is a simple virus. It's a DNA virus. It's got about 8 or 10 genes. It's divided into two regions. In the green are the capsid proteins that make up the virion, and then the internal proteins that are involved in replication, transcription, immune evasion, and things like that are shown in blue. The two genes that many people have focused on are E6 and E7 because these are preferentially maintained and expressed in cancer cells. If you turn them off, the cancers die, okay? And people have been trying to develop drugs against these for eons, and no one's been able to successfully develop a drug against them, in part because they don't have enzymatic activity. So the virion is also simple. As I mentioned, it's composed of two proteins. It has 72 pentamers of L1, which is a major capsid protein that form these beautiful star-shaped objects. And then it's got a minor capsid protein, L2, which sort of inserts, this is like a cutaway inside view, and sticks in here. And then it encapsulates an 8K circular double-stranded DNA genome that has histones. Now, the viral life cycle is actually quite unique as well because the only place it can replicate is in a stratified squamous epithelium. Okay, nowhere else can it generate any viruses. And it's tricky because what it needs to do is it binds the basement membrane, and I'll talk about this a little bit later, and then infects these basal cells where it expresses only the early proteins, the non-virion proteins at very low levels. And it's only when the cells undergo terminal differentiation that it cranks up its genome copy number, circular genome like a plasmid, and starts making these very immunogenic virion structures, as I'll show you in a minute, of being very immunogenic. So it basically hides by basically maintaining ignorance to the infection in the parts of the epithelium where it's under close immune surveillance. So that's why a papillomavirus infection, like a wart, will tend to persist for quite a while. Eventually, your immune system generally will recognize it and get rid of it, but it'll persist for quite a while and shed a lot of virus because it maintains immunological ignorance. This is just histologically and morphologically. You can see this lesion, you can barely even note it. This is on the cervix, actually. And then in the lower levels, you get proteins like E7, or this is actually a protein that's induced by E7. And then the virion-related proteins are only in these upper layers up here. Now, one of the interesting things about HPV-associated cancers, I mean, one of the questions is, why is there a whole lot of cervical cancer and not a whole lot of penile cancer? Because men are just as infected by HPV as women, and they spread it back and forth. And the main reason we think is because at the cervix, there's something called the transformation zone. And that's a place where squamous epithelium abuts simple columnar epithelium. And right in that region, there's like embryological stem cell-like types of cells that for some reason we don't fully understand are particularly susceptible to carcinogenic progression if they get infected. So we see infections throughout the vaginal tract, for instance, but vaginal cancer is actually fairly uncommon. Anal cancer also has the same type of transformation zone. And maybe in the tonsillar crypts, where HPV-associated oral cancers tend to start, there's also this type of epithelium. So it's a very interesting biology that we don't fully understand. So one of the questions is why do the HPV have oncogenes? Well, first of all, they don't have oncogenes to cause cancer because the cancers are sufficiently de-differentiated that they're not virus producing. So causing a cancer is as much of a dead end for the virus as it is for the host. Okay, it doesn't want to cause cancer, but it has a problem because this life cycle I just told you about, in that it's trying to get its DNA genome replicated in terminally differentiated cells, which themselves are not replicating. So it's got to trick the host into thinking, I want to induce replication machinery. And that's what it does with E7 by interacting with several genes. But aberrant proliferation, as many of you know, is a signal for apoptosis. And so it's got to have E6, which prevents apoptosis. And the combination of preventing apoptosis and inducing proliferation leads to immortalization and genetic instability and then transformation. Now, there's a whole cottage industry of trying to understand what E7 interacts with, and there's a whole bunch of proteins, even though only some of them are shown here. But one of the key players is, I'll just point out, is RB. RB is this tumor suppressor that's inactivated in many cancers that are not associated with HPV. In E6, the first, and again, a very key component is that it inactivates by inducing proteasomal degradation of P53, which again, I think as many of you know, is a key tumor suppressor gene that's inactivated in many cancers that aren't associated with HPV. So it really keys into these two controlling mechanisms, one RB that controls DNA replication and RB which controls apoptosis in response to aberrant DNA replication. It's considered the guardian of the genome. Now, in terms of natural history, HPV infections are very common. So about 80% lifetime incidence of having a genital HPV infection is about 80%. So it's almost synonymous with being sexually active. If you're going to be sexually active, you're probably going to get an HPV. But almost all of them go away. About 90% go away within 2 years. Majority within 1 year. But if you get a persistent infection, and if you get a transient infection that goes away fairly soon, that's not going to cause cancer. It's really persistent infection that's on the causal pathway to cancer. And generally within about a decade, you get what we call precancer, a high-grade cervical dysplasia. And then generally, again, about another decade is when the peaks of cancer start to come up. But you note that the peak is actually at about 50, which is actually much earlier than most types of cancers that aren't associated with viral infections. So relatively young women are bearing a very, very high burden of HPV-associated cancers. So I just said this. So HPV is really remarkably quickly taken up. Infections occur. So these are two studies that I've superimposed from the UK in the US in young sexually active girls. And after sexual debut, you can see that almost 50% of them got infected by HPV within six months. And so this is why for these vaccines, which are preventative but don't act therapeutically, it's important that they be introduced into the girls and also boys now before they become sexually active to have the most bang for the buck. So it's kind of ironic that we now have this vaccine, which as I'll show you in a minute, we think is really going to be a great way of preventing cervical cancer. And it's for a cancer that we already have arguably the best screening program, the most effective screening program. I've told you that Pap screening and/or DNA has probably decreased cervical cancer rates at least 80% in the United States. In some countries, they probably do even better. But it's important to point out that this strategy is called secondary prevention. It targets adult women through identifying cervical cytological abnormalities or high-risk HPV DNA, which leads to this big complex algorithm. And eventually, if you have precancerous lesions, you get ablative treatment. And so what we're talking about today is skipping all these steps and doing primary prevention where we prevent the initiating event, prevent the initial HPV infection, which is going to be much more preferable and much better at implementation than screening programs, particularly in low-risk settings. And again, the important distinction is that this targets adolescents, boys and girls. So the vaccine is composed of what we call virus-like particles, VLPs, I'll use that term from now on. And we initially found that if you just express this one major protein, L1, and we expressed it in baculovirus expression vectors and produced it then in insect cells, that this single protein can spontaneously assemble into 360 copies that look for all the world like the outer shell of the virus. But equally, or maybe more important, they not only look like the virus, but if you inject it initially into animals, it induces very high titers of antibodies, very high levels of antibodies that if you take those antibodies out, it can prevent infection with the virus in a tissue culture, or later on we showed in animal models as well. So it not only looks like the virus, it's immunologically and antigenically very similar to the virus. But because the only protein is this one variant protein, they're non-infectious and non-oncogenic. E6 and E7 are the oncogenes. This variant protein is non-oncogenic. So there's no danger of causing cancer with this type of vaccine. And then this, through a number of steps I'm not going to go into, has led to the commercialization of 3 vaccines. And they vary in both the VLP types, the valency, the adjuvant that's used in the production system. Most important probably is the valency. So Cervarix, which isn't used in the United States, is bivalent. It has these 2 high-risk types, 16 and 18, that cause 70% of cervical cancer and 90% of the other HPV-associated cancers. Gardasil, which was the first vaccine, which if you got the vaccine, you probably got that one. It contains the same high-risk types, but also 6 and 11, which don't cause cancer, but are responsible for about 90% of genital warts. So these viruses induce a very distinct disease from cervical cancer. And more recently, and this is the vaccine now that's mostly available in the United States, is Gardasil 9, which includes 5 additional high-risk types that are next in order. I'll show you the order in a minute, 31, 35, and the rest of these. The adjuvant's different too. So Cervarix has a more powerful adjuvant, so it induces higher levels of antibodies. We don't know whether those antibodies, higher levels of antibodies, are critical. So far, the vaccines both look like they're inducing long-term protection from infection. And the production system is different as well. So this is the timeline of the development of the vaccines. And it's just, I think, a good illustration that if you want to develop an intervention, a public health intervention, especially if it's a preventative implementation, you got to be in it for the long haul. So the vaccines, the viruses that cause most cervical cancer, particularly 16, were initially discovered in 1982 by Harold Zerhausen, and he later got the Nobel Prize for that discovery. And it took us 10 years to produce the first VLPs. Actually, when we started to try to produce them, it didn't take very long to produce them, so it took them less than a year to produce them. We didn't think of making the VLPs for 9 years, but then once we started, it actually worked pretty fast. But importantly, during this time that the vaccine was being developed, there was a whole series of laboratory-based studies and also epidemiology studies that led to the proposal that HPV was the first necessary cause of a cancer. Etiologically, it's considered 100% attributed to HPV. There may be a few occasional cervical cancers that aren't caused by HPV, but from a public health point of view, it's almost non-existent. And it's really, really remarkable that here's a cancer that has one primary etiological agent that we can intervene with. You can't say this about any other cancer, not even lung cancer, where smoking is an important component, but people who never smoke still get lung cancer. And this led to coincided with the first clinical trials in 1999. And the first licensure was in young girls in 2006, and then in males in 2009. And one thing to point out is that it was really important that we had a good understanding of cervical cancer progression from HPV infection. That the basic biology really was critical for licensure of this vaccine. And the reason for this is that we couldn't use cervical cancer as the endpoint for a trial. But obviously on the label, we want to say this vaccine prevents cervical cancer, okay? And the reason we couldn't use cervical cancer for an endpoint was twofold. One, you'd have to run the trial for 20 years or 30 years to start to get cancer if what we're doing was preventing initial infection, because it takes that long for initial infection to go on and get the cancer. And the other thing is in trials with active surveillance, we monitor these women for precancerous lesions. And if they get high-grade precancerous lesions, we have to cut them out. We have to cut out the cervical transformation zone. And so we can't let anybody go on to cancer. It's considered unethical. But what we knew at the time is that we knew this natural history of progression. And we knew that if you eliminate, for instance, surgically, these SIN2-3 lesions, women wouldn't get cancer. And so the FDA and other regulatory agencies said, well, if you can show us that you prevent this intermediate and high-grade dysplasias, will say you're going to prevent cancer. So this was all based upon the kind of studies I talked to you previously. And the remarkable thing is, if you look at the most cancer proximal endpoint, cervical intraepithelial neoplasia grade 3, these are high-grade dysplasias. You can see we see 100% efficacy. But it's important to note this 100% efficacy is against incident disease. In other words, they didn't have those HPV infections when they were vaccinated, and restricted to the types that are targeted by the vaccines. The vaccines are type restricted, so they work against 16 and 18, a little bit against a couple of related types, but there's a bunch of types that they don't work at all. But fortunately, you know, those types don't cause as much cancer as 16 and 18. So this actually shows the list of cancer-causing types. So you can see 16 and 18 is 70%. And what Merck decided to do in their second-generation vaccine is add these next 5 types, same type of thing, VLPs. And you can see they're now up to about 90% of all cancer is caused. If you add any other type, you can see you'd only add less than 2% of cancer. So the bang for the buck is really small after those 5 types. And it was a little bit tricky because when Gardasil 9 was being developed, Gardasil 4 and Cervarix were already on the market. And so it was considered unethical to run a placebo-controlled trial where you deny the young women or young men of what was considered a perfectly safe and effective vaccine. And so what the FDA said will give you licensure is based upon 2 things: that the antibody response to the types in common with Gardasil were non-inferior, same level of antibody responses, and they got protection against intermediate and high-grade dysplasia by the other 5 types in a controlled trial where they compared it to Gardasil 4. And what they showed is that there were 96% fewer cases of the new types in Gardasil 9 than in Gardasil 4. So the vaccine was clearly working, giving better protection against these other types. It was a very large trial to have to show this. And so now, right now, Gardasil 9 is the main vaccine that's available in the United States. They pretty much have phased out Gardasil 4. Now, in addition to preventing cervical cancer, there are trials, or cervical intraepithelial neoplasia. There's evidence that both infection and neoplasia at the anus are prevented, also vulvar and vaginal. For penile lesions, there's been trials to demonstrate protection from infection, but not intraepithelial neoplasia, because neoplasia is actually relatively uncommon from HPV infection in the male genitalia. And for the oropharyngeal, again, we don't have any protection against intraepithelial neoplasia because the precursor lesions, which we think are way down in the tonsillar crypts, haven't really been identified yet. We don't know, like, it's completely different than cervical cancer. We know it's SYN 1, SYN 2, SYN 3. Here it's nothing cancer. So we can't do it based upon preclinical disease. So we think that the vaccines remarkably protects against initial infections and induces sterilizing immunity, because most vaccinees never test positive for HPV infection, even by sensitive PCR assays. And if we do see breakthrough infections, they almost always come in the first 6 months or the year of the trial, when the antibody levels are the highest. So why would they come at the beginning of the trial rather than out years farther as you go along? It's because what we're measuring as breakthrough infections are really the emergence of pre-existing infections that we didn't score for, we didn't identify at enrollment. So we did a cervical swab or whatever, and we just didn't see the infection because it was so small that we missed it. And so they all come up in the first 6 months or a year. Then once you get rid of that emergence of prevalent infection, then we get very strong immunity long term. So what the vaccines don't do, and I've already alluded to this, is they don't prevent infection or disease caused by most of the other types that cause cervical cancer, and that's why this arms race to include more and more types. And the other thing they don't do is they don't induce regression of established lesions or prevent progression of HPV-induced lesions. And again, this is why it's so important to be vaccinated before you become exposed to the virus, ideally before you become sexually active. Doesn't mean the vaccine can help you if you're already sexually active, because you may not have been exposed to one of the types. It can still protect you against that type. having one, we actually know from the trials, if you have type A, the vaccine won't get rid of type A, but it would still protect you from infection by type B. And contrary to what you can read on the internet, the vaccine actually has a very strong safety record. It's true that a lot of people have low-grade transient injection site reactions, and once in a while you'll get a little bit of fever. And some of the young people do faint when they get the vaccine, but it's not it's the needle, it's not what's in the needle. So, but it's actually very important, especially in this age group, to sit the subjects down for at least 15 minutes or 30 minutes to make sure they don't faint, because you can break a tooth. That can be a serious adverse event. That isn't really, what's in the needle. It's really it's the needle itself. And this is just some data from some of the clinical trials where if you look at the vaccinees, the people who got the vaccine or the controls, you can see there's no difference in adverse events. And there's been post-licensure surveillance that have looked at this quite extensively. I listed some of the things here. And there's really no pattern of adverse events following immunization in trials or the post-licensure surveillance that would suggest a causal association. There is an issue that autoimmune diseases tend to occur in just about the same age group of young women, like MS and things like that get vaccinated. So there is this temporal association where, there's been 250 million doses of this. A certain number of young girls and boys are going to get autoimmune diseases a day, a month, a year, a decade after being vaccinated. But there's no reason to think that it's causally associated because the rates are not any higher than unvaccinees. And now we've started to get more data from national immunization programs of how the vaccine has been working, mostly in terms of cervical lesions and more in women than in men. And just to show you kind of what the data is starting to look like, these are just sort of snapshots of some data. So this is data of Cervirex, where they look at the incidence of HPV 16 and 18 by age. So this younger group is a group that's going to be vaccinated. This older group isn't going to be vaccinated. And so you can see in the pre-vaccine era, the older group had this level, and it basically stays the same in the post-vaccine era. But in the youngest group that's getting most of the vaccine, when 60% are vaccinated, we had a 52% drop in the population, not just in the vaccinees, but overall in the rate of HPV infection. And by the time 73% got vaccinated, you can see it dropped to 76. So the vaccine is very effective at preventing the infection by the types that are targeted. This is data from Australia and Gardasil. And you can see that in the older groups, looking at intermediate and high-grade cervical dysplasias, there's no change. But in the youngest group, it's dropping off the table. And it looks like there's some data to suggest it may be falling in this next age group where they're getting partial. And we actually are seeing protection against genital warts and cervical infections in women who are not vaccinated due to herd immunity. So a vaccine like this, especially that's sexually transmitted, you get wicked herd immunity. So it's been estimated if you can get 80% vaccination, you basically can eliminate it from the population. And we're seeing quite strong herd immunity even with rates of like 50% in the United States and elsewhere. So that's an added benefit. It doesn't just benefit the people who are vaccinated, but also their partners. And this is just emerging data that there's been an inflection point in the youngest age groups. There's cancer in this age group, cervical cancer, is quite uncommon. So the data is a little bit fuzzy, but it really looks like we're starting to see, this is where vaccine started. We're seeing a decline in cervical cancer rates in the United States, which is really encouraging. We hope that eventually this is just going to drop right off the table if we can get enough the boys and girls vaccinated. I'll talk about that in a minute. So we really have to consider HPV vaccine established products. They've been commercially available for more than 10 years or licensed in 82 countries. People are still thinking, oh, I don't know if I want my kids to get this new vaccine. It's really not a new vaccine anymore. I mean, probably this is a little low. Probably there's over been 300 million doses have been delivered around the world. I mean, that's a lot. And there's increasing evidence for effectiveness, as I've shown you in national immunization programs. So the big problem with this vaccine is it seems like it's really good at what it does, It seems safe, but the problem is implementation, getting it to the people, especially the people who need it the most, which are women in countries who don't get protection by Pap screening. And this is a recently published data that in a non-vaccine scenario, it's expected that over the next 65 years, there will be 19 million cases and 10 million deaths due to cervical cancer, due to HPV-associated cervical cancer, since it's associated with all of them, okay? So the question then is, how many of those future deaths have we so far prevented by 10 years of HPV vaccination program? And you can think of what it is, but I'll tell you in a minute what the data is. And you can look at it as half full or half empty. So it's been projected as that so far we've prevented 365,000 cases and 150,000 deaths. Now, if you're doing a cancer therapy and you said, oh, we just cured 150,000 people of a cancer, everybody would be flying off the table and say, this is just wonderful, you know. But preventative stuff, first of all, there's so much potential that's unmet. And secondly, prevention is underappreciated because the individuals who aren't going to get cancer don't know that they're the ones that would have gotten cancer. It's silent. And whereas therapy, obviously, if you have cancer and somebody cures it, you're going to be off the wall. So it's an example where I think, you know, prevention in cancer and other things as well is really underappreciated. So you can say that, you know, that's a good thing. You can say that we're just getting started. I tend to say we're just getting started. But even, the thing I didn't point out is that the vast majority of these preventions have occurred in higher or upper middle income countries, almost none in lower middle and low income countries, and that's because these girls are just not getting the vaccine. So we got to do something about that. But it's not even just in low resource countries where we're doing a poor job. This is data from the United States, where if you look at the delivery of two other vaccines, the meningococcal and diphtheria, tetanus, acellular pertussis, you can see that their uptake is much higher than HPV, even for a single dose. And so we really need to do a better job of convincing not only parents, but physicians, general practitioners, and pediatricians to recommend the vaccine more strongly. Because clearly there's something happening that when they go into the pediatrician to get these, how come they're not getting these? Okay, there's a disconnect. So how are we going to increase uptake, particularly in low resource settings? Well, both companies have committed to Gavi, which is a big international consortium that buys vaccines to give for free to the 72 poorest countries in the world. And they've agreed to give it to them for less than $5 a dose, which is way better than $175 that we all have to pay. But still, it's more expensive than the entire EPI suite of vaccines. It's still a very expensive vaccine. We're working with manufacturers in emerging countries to manufacture the vaccine. So in India, China, Brazil, they're all moving forward with developing their own in-house vaccines to use in their national immunization programs. As I mentioned, we need to address vaccine hesitancy by developing education programs aimed at families and also healthcare providers. And the thing that we're working on now the most, because it's something that we can do, we're not healthcare implementers, we're scientists. And so we do what we can. And so what we're going to try to do is determine that you don't need to deliver 3 doses. Now, I always forget to mention this, but the vaccines were initially licensed to deliver 3 doses at 0, and then 1 or 2 months, and 6 months. And then it was shown in immunobridging studies that if you're under 15 and you miss the middle dose, you just get the first dose at 0, the next dose at 6 months, you actually respond better than the 15 to 26-year-olds who were in the clinical trials that had efficacy. And so now in the United States and in Europe and other places, if you're under 15, you only need 2 doses. What we want to do is get it down to a single dose. And there's never been a subunit vaccine that's been licensed for a single dose, okay? Because no one thought that this could ever work. But one of the things that we did, so the NCI sponsored a clinical trial. In addition to the two companies, Merck and GSK, sponsoring clinical trials. And there's a lot of discussion whether we should do this or not. And we decided that there was a lot of reasons that we should have a public sector trial. And one of the most important things that came out of this public sector trial is we had the foresight to ask, what happens if you only get one dose? Now, the companies were interested in that because they were looking for licensure for 3 doses. And so, that's what they're going to get licensure. The FDA didn't care what happened with 1 dose. The companies didn't. But we cared for public health reasons. And so, although we didn't have a whole lot of 1-dose recipients, what you can see that even after 7 years, and I can say we now have not quite ready for prime time 10 or 11-year data, is that there's no difference in HPV infection rates for the vaccine targeted types. This is a cervix trial that we did down in Costa Rica between 1 dose and 3 doses. It looks like it actually protects a little bit better, but you can see the confidence intervals are wider because there's actually a quite small number versus here. And importantly, if you look at the types that are partially cross-protected by this vaccine, which are types 31, 33, and 45, the next major oncogenic types, there's no difference in protection at 7 or 10 years. And equally importantly, if you look at the types that we don't see protection against, there's no difference. So why is that important? Because what it says is that there's not differential exposure to sexually transmitted diseases in the 1-dose group, okay? Because one of the complaints would be, well, the 1-dose people, they may be a certain subset that they're just not being exposed to 16 and 18, and that's why it looks like you're getting good protection. But clearly they're getting as many infections by these other sexually transmitted HPVs. So we think that exposures are equivalent. And then once we did those trials, and in some of the company, actually some of the other trials, they started to look at it. And in another Cervix trial, they also found similar efficacy with 1, 2, and 3 doses. And for Gardasil, there was a trial where they were supposed to get 2 or 3 doses, but for political reasons, the trial got canceled or interrupted. And so some of the women only got 1 dose. They weren't randomized to get 1 dose, but some of them happened to get it in some villages. And again, it looks like after 7 years, there's no difference in protection between 1, 2, and 3 doses. So the question is it time to go to a single dose trial? It would just save so much money. It would just be transformative to be able to just go through villages and just give 1 dose rather than have to come back and say, did you get the vaccine? Did you not get the vaccine? Blah, blah, blah. And so should we just go to 1 dose? And we actually think that right now, because these are all post hoc findings, that they're really insufficient to generally promote implementation of single dose trials. In my heart of hearts, I think it's going to work. I fully believe it's going to work, but I think we need to prove it. Now, sort of a hedge bet is you could consider it ethical, I think, at this point, to have early adoptions in low resource settings where they just can't afford 2 doses for everybody. To just give 1 dose with a contingency plan that if it later shows that the 1 dose doesn't work, you could give them a 2nd dose, at least as many as possible, and get that 1st dose into the young girls before they become sexually active. But to put it on really firm footing, we think we need to do a randomized trial to compare 1 and 2 doses, and we're going to do this again in Costa Rica. And this trial has just started. It's a 4-arm trial where we compare 1 to 2 doses of Cervarix and 1 to 2 doses of Gardasil 9. There'll be 5,000 young women in each arm, 12 to 16. And the primary endpoint is going to be persistent infections. So we're going to show that they actually, over 4 years, they do or don't get infection and compare 1 dose versus 2 doses. So we think that this will really give enough proof that regulators will say, yeah, 1 dose is enough. So moving on to why the vaccine works so well. So this is the first vaccine that looks like it's going to be protective by a single dose. It's the first vaccine against a sexually transmitted disease that basically works at all, and it works really great. I showed you that it almost completely prevents infection and certainly disease that's caused by that infection. So what makes these vaccines different, and why do they work so well, and other sexually transmitted disease or subunit vaccines, non-replicating vaccines, don't work in the same way? And we think that there are 2 main reasons for this, and it's been fun to try to explore this and figure out what's going on. And so the one reason is immunological and the other virologic. And the immunological reason is that they are just really good at inducing neutralizing antibodies. And I'll tell you briefly why we think that is. And the other is virologic, in that the way that these viruses infect, which we sort of figured out in a mouse cervical vaginal challenge model, makes them particularly susceptible to infection preventing by circulating neutralizing antibodies. I'll explain that. And then the other is sort of basic biology, because these are DNA viruses. So unlike HIV or hepatitis C, they don't replicate as a swarm and basically be a moving target where they're constantly changing all their proteins. They tend to have a very conservative replication because they use the same machinery as what's used to replicate our own genomes, and so they have a very low mutation rate. And so they're not a moving target that these antibodies have to chase all over the place, like in the case of HIV. And so this really provides a plausible explanation as to why this could be the first subunit vaccine to induce long-term protection after a single dose. So just in terms of seroconversion, this is just one bit of data I chose. And you can see that everybody responds to the vaccine to a rounding error, okay, to all the different types in the vaccine. There's almost nobody that doesn't respond to each of the types in the vaccine. And remarkably, if you look long-term, this is out to 10 years, you can see, especially in the target age and younger, 15 to 25, that you see a peak after the second dose, or the third dose in this case, and then you get a plateauing where basically it flattens out and doesn't change. As long as we're able to measure, once you reach that plateau, those antibodies just keep there. And so we think that what's happening is it's inducing what are called long-lived plasma cells, which are cells derived from naive B cells, which when they get a really strong signal, go to the bone marrow and essentially sit in a bone marrow niche and pump out antibodies for the rest of your life. This is what you see after a virus infection, let's say a measles virus infection, which you guys don't get, but we all got. And so it's more indicative of what you see after a real virus infection than of a protein-based vaccine. And the remarkable thing is that if you look even after one dose, we see the same plateau effect, where this is 7 years, but now we even have 10-year data where it's absolutely flat. There's no change in antibody responses after year one. And so this is why we think we're going to get long-term protection, because we're getting long-term antibody responses. If you look at a tetanus vaccine, they would have a peak and then go drop down. But here we're getting maintenance of antibody responses. And again, remarkably, if you give 3 doses, the levels of antibodies are only 4-fold higher than if you give if you give 1 dose, if you give 3 doses, it's only 4-fold higher than if you just give 1 dose. So you don't get that much added bang for the buck. It's almost like that each dose is additive. And it's still 9-fold higher than after natural infection. Now, the reason we think that, the simplistic reason we think that these vaccines are so good at inducing antibodies is what they do to the B cell receptors on the surface of naive B cells. So when simple monomeric antigen, say tetanus toxoid, sees these immunoglobulins that make up the B cell receptor, they don't cross-link. They just get relatively weak signals, which results in low antibodies that don't last a long time. But if you have these repetitive elements like this, we think that what happens is that you get very strong signals because it cross-links these receptors and then activates these tyrosine kinases, which are tightly associated with these B cell receptors, which sends very strong survival and proliferation signals, resulting in high antibodies that last a long time. Basically, strong induction of long-lived plasma cells. And we think that this biology makes sense and has evolved because this is the way that B cells recognize something that's a foreign, a microbial surface versus one of your own proteins. And that there's a special spacing of 50 to 100 angstroms that are found on viruses, they're found on bacteriopilli, they're found on the surfaces of a lot of parasites. And this epitope spacing like this is not found on surfaces of the vertebrate animal that are exposed to the systemic immune system. So basically the B cell receptor is like an antigen-specific pattern recognition receptor. This is how it really distinguishes foreignness from self in part. So moving on to the virology, the reason that we think that the antibodies that we generate, so I told you we generate a whole bunch of high titer antibodies, And these antibodies, we think, work really well because this is the slowest infecting virus of all time. It's really interesting because it can't bind the apical surface of the epithelium, even like a simple columnar epithelia. It just floats around, doesn't bind anything until there's trauma to expose the basement membrane. And then it binds to specific modified forms of heparan sulfate proteoglycan unique to the basement membrane. They're not found on the apical surface of the cells. And then this induces a conformation change in the virion that exposes the minor capsid protein, which is tucked in here, to cleavage by a protein convertase called furin, which then moves this minor capsid protein out of the way and exposes the receptor binding site for the keratinocyte, the basal keratinocyte. And the important thing is that all of this takes hours and hours, okay? And so there's a lot of opportunity for antibodies to prevent this whole process along the way. And I'll show you some of the ways that we think it works. And the other important point is that now what this allows is that the antibodies that can protect can be systemic antibodies. They can come from the capillaries or the interstitial spaces and just ooze out, be exudated at these sites of trauma, so that the virus is trying to infect against an outgoing gradient of antibodies, such that probably close to the basement membrane, the levels of antibodies are actually quite similar to what they are in the interstitial spaces. And so that's why we can do systemic immunization, which is really good at generating systemic antibodies, but really bad at inducing mucosal IgA. Okay, that's why we think the vaccine can protect so well, even after systemic immunization. So again, we got really lucky. We didn't know this at the time, that this is what was needed for the vaccine, for the virus to infect. But it's been very interesting to figure it out. So one of the questions we asked is are the plateau titers after activation near the minimal level needed for protection? And will this fourfold difference between antibodies levels after 3 and 1 dose influence long-term protection? And so to do this, we did what was called a passive transfer study. So we have a mouse surgical vaginal challenge model where we take what are called pseudovirus. So instead of the real genome, we package luciferase expressing plasmid, and we actually then infect them intravaginally after trauma with a non-ionic detergent. And so what we did, we took rabbit sera and diluted it and put it into a naive mouse who hadn't been vaccinated, and then challenged these after the antibodies had a chance to circulate around. And we asked the question, well, how much can we dilute these antibodies and still get them to work? And this was one of the most surprising things that I have ever had in science. So what we found is that even when you diluted 10,000-fold, so that the circulating antibody levels in that mouse that was protected from challenge was 10,000 less than in that rabbit, we still got protection, okay? Now, rabbits generate about 10 times higher levels of antibodies than either mice or humans. So we would say that probably in a mouse or a human, you could dilute it about, that they probably have 1,000 times levels of antibodies than you need for protection. So from this, we speculate that that fourfold difference between 1 and 3 doses isn't going to make a whole lot of difference if you're 1,000 times over what you need. And that's probably why we're seeing such strong protection after a single dose. And so in a reflection of this, if you look at how much the antibody levels that are needed to protect in an in vitro neutralization assay versus protect an animal from cervical vaginal challenge, you need about 100-fold less sera to protect in the in vitro assay than in the in vivo assay. So we think that the in vivo assay is missing an important mechanisms of infection inhibition. And what we think is happening is that at high levels of antibodies, you're just preventing, and we actually have evidence for this. We can look in the vaginal tract. I won't show you the data, to show you the cartoon. Is that higher levels of antibodies, the type that prevent in vitro infection, we basically prevent binding these HSPGs. At low levels of antibodies, we actually get association with these HSPGs, but they never get onto the cells. And so we think it happens is that these antibodies, when they're sitting here for hours, act as handlebars in monocytes and macrophages and neutrophils, which are also attracted to these sites of trauma. They just gobble them up, okay? This is a signal for those guys to phagocytize these viruses. And so they get removed before they have a chance to undergo all these conformation changes and bind the cells and get internalized. So to conclude this portion, I think I have time. The HPV vaccines are very effective at preventing incident infection disease by the vaccine types because the VLPs are exceptionally potent inducers of neutralizing antibodies, and the virus is really susceptible to inhibition by antibodies. And so the vaccine has great potential for reducing the burden of HPV-associated cancers worldwide. And the big thing now is to just get it to the people who need it. And we think that demonstrating sustained efficacy in a single-dose randomized trial could transform implementation programs around the world. So in the last few minutes, I'm going to tell you a little bit about translating, this is translational oncology, what we found about how these viruses infect and translate it into a cancer therapy. And again, we didn't start out to try to do this. This is just following our nose and finding out what happened. And so what we had, recall that I told you that the VLPs or the virus can't bind or infect normal tissues. But what we found is that because they don't have these heparin sulfate, these specially modified forms of HSPGs that are restricted to the basement membrane. But come to find out, almost all tumors have those same types of modified HSPGs. So the reason we think that this has evolved is because when you think about epithelial cells, where do they normally divide? When they're attached to the basement membrane, just the lower level. And so what we postulate is that they're getting critical signals to divide and not differentiate from these HSPGs that are restricted to the basement membrane. And now when you have a tumor and you're off in space and not on the basement membrane, you still probably want those signals. So how do you get them? You basically induce those modifying enzymes so that either the extracellular matrix you generate or the cells themselves now have those modifications. And so now basically what we found is that HPVs can bind and infect a wide variety of tumors, certainly most epithelial tumors and melanomas. And so the HPV-VLPs can then be essentially guided missiles to deliver cytotoxic agents to tumors. And this is just an example of, if we take a virus and we label them with a green dye, so where they bind is green, you can see if you put it on undisrupted tissues, they don't bind at all, they just wash off. But as soon as you disrupt it, in this case with a non-ionic detergent, it exposed the basement membrane, you can see the basement membranes line up like a Christmas tree. And this is true of epithelia, but also mesothelium in the peritoneal cavity. So they also have those same types of modifications. But if you look at just the opposite, if you look at tumor cells, you can see that this is just an example of tumors from various sources in various cell lines, is that if you again use these label particles, and this is a FACS plot, you can see that all these tumors bind strongly. And the reason we think that this is involved in binding the HSPGs is you can block it with heparin, which is just a highly sulfated form of HSPGs. And if you treat the tumors with heparinases, then they no longer bind, okay? So that's why we think that it's really HSPGs that all these tumor cells are binding. So this is just an in vivo example where, just to show the co-localization, the dramatic co-localization in infection in this case. So what we did is we did IP injection of these SHIN3 cells, which express the RFP gene. So where the tumors are is red. You can see if there's no tumors, there's no red. If there's tumors, there's red. And then after a few days, after the tumors grew up, we did IP injection, not tumor injection, just IP injection, just let it float around, of pseudovirus that express GFP. So the infected cells in the peritoneal cavity will fluoresce green. And what you can see, appreciate probably best down here, is that there's exact co-localization of the two. So you can see that all these little tumor nodules are now lighting up. You can imagine that you could identify where these tumors are, because ovarian tumors in people don't fluoresce red. So if you wanted to identify them, you would infect them with these pseudovirus and you could identify the tumor nodules. So you can imagine using this for either with radioactive or dye doing imaging, or you could deliver cytotoxic drugs. But the thing that we're concentrating on is direct killing using a dye which is called IR700, which is excited in the infrared. And when this dye is directly opposed to a membrane, it pokes holes in the membrane, maybe in part probably by free radical oxygen generation, and the cells just shrivel up and die within minutes. And so what's really nice about this for cancer therapy is it's got dual specificity. So normal cells don't bind. Only the tumor cells bind. And only where you shine the IR light do you get killing. And so this way, there's going to be very little off-target effects with this type of treatment. And so we got to thinking, well, what would be a really good cancer to try this out in people? You know, what's a cancer that you really don't want to have off-target effects and you really want good specificity? And you may think of, maybe you got better ideas than us, but this is what we decided to do, is ocular melanoma. Because ocular melanoma is a real problem because the ophthalmologists see it, but it's hard to distinguish whether it's just a nevi or a real dangerous melanoma. And oftentimes they wait too long and then it's already metastasized and people die of liver mets. And the reason they wait too long is that the only thing you can do is either take out the eyeball, which is, you know, has some adverse side effects, or they can put radioactive crystals behind the eye. They have to do an operation, and then they spew out this radioactivity, which not only inhibits the tumor, but also makes them blind. And they're also spewing out radioactivity all this time they're walking around. So clearly there's a need for something that's specific. And the problem with this is that the tumors tend to form in this choroid space, which is right behind the retina. So you have to do something that can kill these cells without hurting this, which is right up opposed to it. And so the nice thing about this cancer for this kind of treatment is that you can see it and you can shine the laser through the vitreous right on the tumor. And another good thing is there's a rabbit model that's been developed for, that involves this intrachoroidal injection of human ocular melanoma cell lines. And we developed this project in collaboration with Oro Bioscience in a CRADA up in Boston. And so in this rabbit model, what happens here is that you have to cyclosporin treat the rabbits because these are human, you know, melanoma cells, and so you have to immunosuppress them. And what you do is you let the tumors form, and then you just do intravitreous injection of the dye-coupled particles, and they just float around. And again, the only thing they bind to is the tumor cells. And then 6 to 8 hours after you do that, you shine for 80 seconds this infrared light. And if you do this twice, after 2 days, you can see there's massive necrosis in a dose-dependent fashion in the tumors. Okay, this is an untreated tumor. It's actually bulged out of the eyeball here, but you can see there's massive necrosis. And what's really impressive, if you wait 8 or 9 days after the second treatment, the tumors have basically melted away in the 20 and 50 microgram, and there's no evidence histologically that there's any damage to the retina. So this allowed us, and the FDA agreed, to go on to a clinical trial of this idea. And I have to tell you, it was really scary when we injected into the eyeball this first. I didn't do this, of course. These are the people at Wilms Eye and all that. But to have these people get injected with this dye-coupled particles and shining the laser was pretty scary, you can imagine. But the patients are all for it because they don't want it. I mean, the other treatments are so bad. And so this to give you an idea, this was a typical dose escalation. So we increased in a dose-wise fashion from 20 to 40 to 80 micrograms, the frequency of treatment, either 1, 2, or 3 weekly injections. And in the end, we actually went from 1 to 2 applications of the laser that was separated by 30 minutes. So you can see this is typical. You do 1 dose of the lowest, and then you go up, then you start doing multiple doses, and then you do multiple doses, and then you do 2 lasers. And I'm happy to report that to date, we've done 24 patients. There's been no adverse events, no dose-limiting toxicity. There's been no loss of visual acuity in anybody. The patients are all fine. They don't even feel anything when you do this, okay? So there is a little bit of inflammation that's seen. It's interesting, this inflammation comes in 2 to 4 weeks. There's not like immediate inflammation, like it's an innate response. We really think this is an adaptive immune response to these cells dying and spewing out their guts, which is a very immunogenic type of death. It's actually a very positive thing that we can control just with like topical steroids and that. But we actually think that this is part of the reason why, so far, only one of the patients at the very lowest dose has gone on to standard of care. The other ones either have had reduction or stable disease. And so some of them are out for more than a year. And now we're moving on to a phase 2 trial where we're going to use the highest dose, 80 micrograms, 3 times, 2 lasers in a group of patients, and hopefully within a year get into a phase 2 trial. So it's an example, I think, where if you do some basic biology and sort of figure out how one thing's working, you know, one intervention, this was a prophylaxis, you can actually end up with something that can lead to a cancer therapy, hopefully. Okay. So the most optimistic projection for this topic is that this could be a broadly applicable off-the-shelf cancer therapy. And so now the question is, what cancer is next? So if you have any ideas, you can tell me. But I'll tell you what we're thinking about. We're thinking about bladder cancer and head and neck cancer are the 2 next things we'd like to go to. So just real quickly to thank all my many collaborators, you know, especially Doug Lowy, who's been my partner in crime in the LCO for about 30, 35 years. People involved in the clinical trial in oral bioscience, Ellie de los Pinos, Rhonda Kynes, and Steve Monk, who are very closely involved with the clinical trials I just mentioned. Thank you.
Questions for John?
Speak loudly.
This is like more of a public health question, but one of the reasons I've heard patients, specifically in the U.S., not want to give their children the HPV vaccine is they think they might be young and then make you more promiscuous. So I'm wondering, I know it's absurd, but I'm wondering, are you able to have a secondary endpoint of like number of partners, for example, in that clinical trial you're doing for like 1 or 2 vaccines, and maybe compare that to public data in general, just to disprove it?
Well, you could look at it. You could potentially look at it secondarily, but obviously they're blinded, so you couldn't do that. But there has been studies that have looked specifically at that question, not in controlled clinical trials, but just in general, you know, in national immunization programs. And there's absolutely no evidence that it influences sexual activity. Hey, if you're not afraid of HIV, there's no way protection against HPV is going to make any difference to you. So no, those fairly reasonable studies have been done, and there's no indication. And we didn't expect it to be, but that's not what's deciding whether people are going to have sex, because they're afraid that, you know, in 40 years they may get cervical cancer. Yeah. Can you speak up a little bit?
Sorry. How was the age cutoff decided, and how were the why were boys excluded to get from the general population?
So the age was set up because you wanted a fair number of the women to not have HPV infection at entry, because we're looking at this as a preventative measure. But you also wanted, in a 4-year trial, a lot of them to have exposure to HPV. And women in the, you know, say the 16 to 26-year-old is an age where they're likely to have more new partners than older women. And the chances of them already having been exposed goes up in older women. And that means 2 things. Either they already have an infection, or they've had an infection and they've cleared it, and so they're not going to get reinfected. So the number of events goes down as women get older. And the reason they didn't go lower is because to get informed consent, you start getting too low, then you really have to involve the parents in informed consent, and it becomes much more difficult. And you'd have to, again, wait longer for them to have their events. So it was just sort of a practical consideration. In the boys, the reason boys were second was, it was just, these were big trials, 20,000, you know, people trials. And it was thought that because most of the burden of HPV-associated cancers is really in women worldwide, that it made sense to do women first, okay? And the other thing is that we understood the disease process much more, okay? And we could use these clinical endpoints, these high-grade dysplasias for women, but as I said, for oropharyngeal cancer, all we would have would be HPV infection. And the rates of carriage in oral cavity is 10-fold lower than it is in the cervix. So even for infection, we'd have to have 10 times bigger trials. So that's why.
Follow up with the why now. Is there a blood test that you use for antibodies before they give it, or does it matter?
No, because one is that not everybody who is infected or has been infected seroconverts, and almost nobody would have antibodies to all the various types. It's just not cost effective to do it. Those are good questions. Yeah.
So in the cervix, when it changes from the columnar epithelium to the squamous epithelium, so are the chances of basal membrane being exposed there higher? That's why it's higher.
It's not that the infection rates are higher. It's just that if you do get infected there, the chance of that infection persisting and going on to cancer is higher. Okay. It's not that they're especially susceptible to infection, as far as we can tell. Like I say, infections in the vaginal tract and other places in the cervix is quite common. It's just that they tend to go away and they don't tend to persist as much. And the main thing that even given persistence, they're much less likely to go on to cancer than at this special transformation zone cells.
And are there other cancers where such cases where epithelial transformation occurs from one type to another, like in colon or in gut?
Yeah, there is, yeah, so for instance, like Barrett's esophagus is another term for this is metaplasia, where they're sort of going back and forth between these two forms, squamous and columnar. And in Barrett's esophagus, which is a risk factor for esophageal cancer, is really an abnormal metaplasia where there's more of these transformation zone kind of things. Now, that's not been associated specifically with a viral infection. But so, yeah, transformation zones may be just because of this remodeling and changing between back and forth, that plasticity maybe increases the chances where things can go haywire. Probably initially epigenetically, I would imagine, you turn on different epigenetic modifiers, and this can change, go wrong a little bit more. That's just a guess. I mean, it's not. All right. Thank you.